Such an honor to be here. I see myself in each and every one of you because I was a transfer student. So yeah, we can make it. Before I go into my project, I'm just gonna introduce myself and talk about my journey in the STEM a little bit. So as Rance said, my name is Jocelyne Fadiga. I'm from the Ivory Coast. Who knows where that is? Where is it? Because a lot of people say, yeah, I know, I see. And then when I ask, it's like, where is it? No. So it's on the African continent, on the west coast, and it's next to Ghana. But unlike Ghana, it used to be a French colony, so the main language there is French. So I moved to the United States in 2008, on December the 15th of 2008. And when I came, I had no English proficiency at all. I was only speaking French. So I went to ESL courses, took a bunch of writing courses, grammar, and then transferable courses. And at first, I wanted to study engineering. And then when I transferred to UC Merced, I transferred as a material science and engineering major. After my first semester, I had good grades, but I wasn't able to see myself in the future in that major. So I decided to change it. And I knew that I loved math and chemistry. So I changed my major. I majored in chemistry and applied math, and then a minor in writing. And I graduated last May. So I'm really fresh. So keep that in mind when you ask your questions. I don't know a lot. I just started at UCSF in June, so it's only been 5 months. So yeah. And feel free to ask questions as we go through this. Okay, so my project is about characterizing or defining cell identity in an epithelial stem cell lineage. If you are a bio major, you probably know what I'm talking about when I say epithelial or epithelium. But in essence, it's the layering cells in a tissue. It's like the surface. So I'm trying to define what stem cell divide into, what they produce, what their daughters are like, what type of differentiation process happens. And in our lab, my professor, his name is Todd Nystul, and in our lab we study the fundamental properties of epithelial stem cells, their associated niche. A niche is the environment where the cell lives. So their associated niche and the connection between epithelial stem cells and cancer. As you can imagine, if a cell, like cancerous cells are cells that proliferate, that divide, but at a much faster rate than normal cells. So understanding stem cells and being able to manipulate how they differentiate can help us find cancer therapy and cancer cure. So that makes me excited because it's something very real. My project in the scope of the Nystul Lab project is to, like I said, characterize cell identity in the epithelium. So why do we feel like why is my project important? Why am I trying to define epithelial stem cells? Because after decades of study, we still don't know what makes a stem cell, what gives a stem cell its stemness. If I'm a cell and I'm dividing, I make exactly a copy of me, so a prototype. But a stem cell makes another cell that can become a skin tissue, an eyeball, bone, blood cells. So how, from a stem cell, do you become something completely different with different properties and different potential. That's why, and after years of studies, we still don't understand that. So that's where my project comes in. The molecular basis of stem cell fate is not well understood. How from the stem cell, like I was saying, you become, a cell becomes a blood cell, for example. And gaining understanding in the cell identity and their lineage can help us more accurately interpret our experimental result and has translational potential. The aims in studying cell identity is first to determine the number of active follicle stem cells, FSCs, and other cell per germarium. I will define what a germarium is in the later, the future slides. Aim 2 is to describe the diversity of expression patterns in the early FSC lineage. So what I mean by that is, when I have, so we study Drosophila, which are fruit flies, and when we dissect them, we take out their ovaries. They're like very tiny, and then at the tip of each ovariole, which is, so if you think about like a set of grapes or bananas, they're all connected, right? The ovaries are like that. And each banana is an ovariole. And at the tip of each banana is the germarium. It's a very transparent, very tiny part of the ovariole. So what we do when we dissect them, we stain them with different type of stain, histology, to either highlight a part that we want to study. When we look at it through the microscope, we'll see probably maybe the membrane or the specific cells or the stem cell itself, stuff like that. So this expression patterns is, if I'm staining my germarium with something like a membrane stain, I want to see how far it goes and how intense the stain is, and which cells have the stain, which don't have the stain. So that helps, making a comparison or establishing a difference between cell A and cell B. Aim 3 is determine the identity of FSC daughters that participate in FSC replacement and competition. Because in the cell environment, there are instances where one cell makes a daughter and the daughter comes back to replace the other mother. And we want to understand how that happens. Because if you think about cancer, they proliferate, right? If cancer in the scope of competition is hyper competition, right? If you understand how competition happens in the environment of the cell, you can probably stop it, right? So we also study cell replacement and competition. Yes?
So we're talking about dissecting the ovaries out of the Drosophila.
Yes.
The Drosophila are so small.
They are,
so
yeah.
What special tools do you have to use to get the ovaries out?
So we have microscopes that are like really, they zoom in. And so first of all, let me just be like completely, what I do, they're in a vial. Before I dissect them, I want them to be plump, right? And I keep the female with the male because when the male are in the vial, the female thrive. Kind of like, like people. So I keep male and female in the same vial. And if I'm dissecting, let's say Friday, starting Wednesday or Tuesday, I put wet yeast. They love it. Yeast, you mix it with water, and then you put a little bit on the wall of the vial, and then you keep them in. And every day you change the vial, and you put fresh wet yeast. They eat the wet yeast, and the ovaries become huge. So they're a little bigger, and you can differentiate. It's very easy to differentiate female from male. The female are bigger because they have babies, ovaries. So from the tip of the abdomen, I pull with a forceps, a tiny forceps, looking under the microscope. So I'm looking, and my hands are under. I pull the tip of the abdomen, and then I rip the tissue, right? and slowly I squeeze from the middle of the abdomen, and then the ovaries come out. It's really satisfying. It's really satisfying. So what are some of the tools? As we mentioned, dissections, tissue staining, confocal microscopy. So confocal microscopes are like very powerful microscopes, and that's what we use because we want very high precision and accuracy since we're trying to describe stuff. And machine learning. So we are collaborating with a group of people, 2 people actually from Caltech, and they're writing codes for us to identify specific cells. So if we see something repetitive, a pattern, if cells are looking the same, you can just write a code instead of going through the same process over and over again. So machine learning is in collaboration with people from Caltech. And image processing via Merced. Merced is a software with which you can visualize your images 3D. So when I image I put the ovaries on glass slide, right? When I image them, I make layers of the image. So Z-stack, and they're like to the level of microns. So very, very thin Z plane in the image. And when I take that file, sometimes there are 1 gig of one image. When I take the file and I go to Merced, Merced puts all the Z-stacks together and gives me the whole ovary, like in 3D. So I'll show you some videos. And of course, I forgot. Clonal analysis. So we induce clones into the ovary to track their path sometimes. And here I'm showing male and female. So the female is on the left and male on the right. Male usually have like a dark back and they're very small. You can tell them apart easily. Yeah. Feel free to interrupt if you have a question. And these are the ovaries. The translucent part right there are the germarium. So that's what we study. So this big thing is not necessarily of interest. Yeah. Okay. So here we have the ovaries, like I was showing, and one ovariole. Zoomed in right here is the ovariole, and at the tip of the ovariole we have the germarium. So these are terminal filament cells. These are cap cells. Escort cells are sort of cells that help the other cell. They're almost like babysitters. And the FSC, we have one here, another one here. In our lab, and probably I think a couple of other labs, we have defined 2 stem cells per germarium. But there was a recent paper, I think in 2016, a lab at Columbia University said that there are 16. So you understand why we need to come to a consensus as far as how many stem cells we actually have. And the GSC is germline stem cells. Yeah. Yes?
Are the stem cells only in the germarium? Is that the only place they're found in the entire ovary?
Yes. Yes. Yeah. So I was talking about staining, right? Some of the stains that we use are disc large or DLG, which is a membrane stain. Fascicline 3 or FAST3, also a membrane stain. Castor, which stained stalk cells and polar cells. So stalk cells are here. These. Between each follicle, between each round part, you have stalk cells. And that's what castor stains. And I is absent, or Aya, stains main body follicle. So one of the big round figure is stained by eyes absent. And of course, you probably know GFP, green fluorescent protein. This can be target specific. So sometimes, if I want, for example, I want to see a specific set of cells in a specific part of the germarium, we will drive GFP into that part of the germarium. And we have lines of flies. We can cross them, they make babies, we take the F1 generation, we stain them with GFP, and we can see GFP under the microscope in that specific part, if you made your cross right. So one of those drivers, we have 10930, 13CO6. Those are just names that I found. So we call them that, but there are genes that people have identified, and they target a specific part of the germarium. So GFP is very useful. You can just target any part depending on the cross that you made. VASA, it's an RNA binding protein, and usually VASA stains the germline stem cell cytoplasm, so anything but the nucleus. And 4,6-diamidino 2-phenyl-indole, or DAPI. DAPI is easier. That's a nuclear marker, and it stains just the nucleus, all the DNA inside the cell. So these are some of the images that I made, or I took. And the one on your left is 13CO6 Gal4, which is a driver. A driver is, so it's on the genome, you can have pieces or promoters of genes where you will put something like Gal4 to drive a specific expression or to overexpress the gene. Gal4 is one of those things that you will promote you will put on the genome, on the piece of the gene to promote, for example, in this case, GFP expression in this part. So 13CO6 covers that part of the germarium. So everything that's green is GFP. Everything that's red is FAS3. And I was telling you it's a membrane stain, right? So you see it's not touching anything like any round ball. The round blue is DAPI. So all of that is nuclear. And DAPI is, so have you heard of DNA intercalation? So you have sometimes molecules that, benzene does that, molecule that go between sheets of your DNA, and that's what DAPI does. So the DNA is helical, and the molecule goes in between, and you can see the blue when you look at, with ultraviolet light, when you look at it, it becomes blue, pretty much. And then the one on the right is C587, which is another line of flies, and CD8 GFP with also FAS3, membrane, and then nuclear DAPI, and all of this is GFP. This is another image. Oregon R is a wild type fly. So those are what I would call normal flies. We didn't manipulate them. This is just how they are in nature, and that's one of the ovarials. And remember I was telling you about stalk cells. These are stalk, and this is castor right here. Everything, so when the germarium evolves, it buds, right? And the budding grows and becomes an egg, or a follicle and then an egg. And then the egg comes out as a pupae, and then the pupae grows into a fly. So the evolution of a fly starts at the tip of the germarium, and then the budding This part right here is where the budding starts. And then this is going to grow into something like that, and then something like this, and then eventually a big egg, and then a fly outside of the, yes. Why did it choose? That's a good question, because they're very simple. Because they're very simple. So when you wanna study anything, you start with a simple system and then you extrapolate. You don't want to start with the complicated. It will be hard to. First of all, you probably will it will take forever to find anything because the system is so complicated. So flies are very simple. They have a short life expectancy. They last 3 weeks. For probably like maybe 40 days at most, but and then they make a lot of babies, and it's cheap, cost efficient. Yes.
Can you repeat the question?
Yes. So her question was why did my professor choose flies? And then I was saying because it's a simple system to study. It's cost efficient. They make many babies, so we can keep on studying them. They have a short life expectancy. Can't talk, and yeah. So earlier I was mentioning Ovaries, which is the software that I'm using to analyze my images. A lot of things that I do take me hours. After taking my images on the microscope, I spent hours staring at a computer. And what I do is, because the stain, it's when it's on 3D, it's very fuzzy. You can't do anything. So to isolate every cell, I cover it with a surface. And Ovaries helps me with that. So this is a video of the process that I go through. The green again is GFE and the red is FAST3. The blue is the nuclei. So now you see that it looks solid, right? It looks dense because I covered every nucleus with a surface. And then I covered the green channel or GFP with another surface, and then the red with a surface. It looks very simple, but it took me like months to actually get the correct parameters and things like that. So why did I do that? Right now, in one of the techniques that we're using is to try to identify how many cells are both expressing FAST3, which is red, and GFP, which is green. And I found that there is a region of overlap here in the middle, and that region is called 2A, 2B. In that region, we want to know how many cells both have green GFP and red FAST3, and count them specifically. So what I did was covering each channel, which is like the fuzzy filter that we saw, into a surface, cut up the surfaces into pieces, and then plot that with Excel, because I get data from this software, so I use the data and I plot. This is another analysis that I'm doing. So this is viscon GFP, and viscon GFP is also a line, but in the germarium or in the ovarial in general, when cells divide, they leave tracts, and those tracts are called ring canals. They're actin rich, and they're between each cell. Like if I divide, I will have, and I give you or my daughter, I'll have a we'll have a ring canal between us. So viscon is a protein trap, and it's an identifier of ring canals. But when you stain the fly with GFP, every ring canal in the ovarial will be green. So it's a way for me to, for example, if I see a ring canal between two cells, I know that there was a division that occurred. So I can track the cells. And if I see, for example, in this follicle, two groups of cells, and they're not affiliated, they're not connected with each other, then that would tell me what something like they're coming from one mother each, right? If I see 4, then I probably have 4 mothers, right? So this is a technique that we're currently using. It's very, very in its like preliminary stages, but we're currently using it to track cells and see their lineages, where they're coming from, and if they're sisters. Yeah, so I'll show you the process. So I picked the follicle. That one is stage 2 because it's simpler. Every nuclei is discrete, and it's easier to track since I'm starting. The red was the membrane marker. Let me see if I can pause. So I wanted to pause to sort of explain a little bit what's going on. When I pick one spot on that follicle to start my analysis, and from one cell, I track down the next one. If I see a ring canal, a green shape between those two cells, until I find maybe like 5 cells. When I find 5, I change their colors because I don't want to recount them. So that's why they're a fuchsia or magenta. And I do that in groups every time until I cover the full surface. So for this specific follicle, I didn't find 2 distinct groups. Maybe I'm wrong. So I'll probably do this again. Yeah. The red is the membrane. The membrane is sort of to double check if I'm, tracking them correctly, because I would have a ring canal embedded in the membrane. So if I put back the membrane and I see that the ring canal is in the membrane, then I track them correctly. But if it's not, then maybe the sister is the wrong one. So when I cover the whole thing with surfaces, it looks like this. You can see castor here, here. So these are probably going to become like this because they're expressing the green castor. And the Aya that I was talking about, or Aya is absent. They're called Aya because when they found this specific protein, the flies didn't have eyes, so they call eyes absent. A lot of originality. So the eyes absent is red, and it covers the follicle cells, and sometimes it goes all the way to like region 2A, 2B, like I was mentioning. And this is just a bonus. This is an image of viscone, and you can see the ring canals. See that? So during growth, cell growth and division, or after division, the cells share matrix, protein between each other through those ring canals. So it's like a communication medium. And I'm just showing you pictures of what my clones look like. So to put clones into, negatively marked clones into an ovary or an ovariole, what we do is put the flies under heat shock regimen. One funny thing is that a lot of times when you heat shock them, you put them in a water bath at 37 degrees for an hour. You can do it 2 times, 4 times. Commonly we do it 4 times. So twice twice a day for 2 days. When you heat shock them, the male die quicker than the female, so just putting that out there. So in this specific fly, we have, my heat shock regimen was 1 hour, so I only heat shock them for an hour that day. And then right after the heat shock, you feed them every day wet yeast for 7 days typically, but I did it for 9 days. I was just starting. So the next one is just 1 hour times 4 for 4 days. And then this is 1 hour times 2 for 9 days. So I changed the regimen because I just wanted to see how heat shocking was affecting the clone behaviors and stuff. So what I want to do with what I have so far is apply the 13C06 that I was telling you about that shows GFP in the 2A2B until the tip of the germarium. I want to use that and also add a combination of antibodies and do the analysis. So I don't have the data because it's very new, but I want to apply the analysis that I did on 13C06 on other combination of antibody to see where there is an overlap. And I also need to complete the analysis of the Viscon GFP, the video that I showed you, and induce a clone in Viscon GFP that way. So the clone usually marks stem cells. If I have a clone in Viscon GFP which shows ring canal, I can clearly see which color of stem cell make what type of cell, later on. So yeah, that's pretty much it. And this is my lab. From left to right, left we have Sumitra, she's a grad student. She's a 4th year grad student. With the UCSF hat, we have Marimar. She is also a research assistant like myself. And then me, and the postdoc on the far right is Rebecca Kim. She just had her PhD last month. And she's doing a postdoc in the lab. And here is my PI and his kid. The guy on the tree is my PI, Todd, and then 2 of his kids. Yeah. Oh, his 2 kids, actually. He doesn't have anymore. All right. Thank you so much. And questions? What do you want to know?
So,
yes.
So you said that you got your undergrad in chemistry and
Yes.
applied mathematics.
Yes.
And now you're doing this. So can you explain a little bit how that happened?
That's a good question. So I actually didn't plan this. Going into Drosophila, I have very little biology background, and in my lab we even use genetics, and I did not know any genetics at all. I think the reason why I joined the lab is the PI. Yeah. And I would say that's so important. I don't know if you guys are like me, but I need to feel like my input is valuable and appreciated. And that makes me perform. And the PI offered that. So I met him when he came to UC Merced at the time, and he gave a talk on his research. I went because my friend texted me and told me there was food, so I ran. You know how it is. So I ran there and I went, but I was interested in his research, and I didn't even plan on applying to UCSF. But when I got there, he talked about graduate programs at UCSF and research. I was like, oh, maybe I have a chance. And after the talk, I talked to him, and he was like, yeah, you should apply. And I applied, but I did not get it. So what I did was, like I sent him an email and said, I didn't get in, but I'm not discouraged, I'm gonna apply again. And he called me, he emailed me first and he said, you seem to be very interested, I can send you papers to read and then we can talk about it. So he sent me a bunch of papers that I read, religiously and marked all of my questions. I had like 3 pages of questions. And we talked on a Friday and then we talked about my questions, the research, and he decided to take me on. So we wrote together a grant and then we submitted the grant to the NIH and the grant got approved and then I started. Yeah, so this was a good, lucky turn of event. Yeah, but I didn't necessarily plan on going into Drosophila. Yeah. So be open. I guess that's the moral of the story. Be open. And for grad school, you don't necessarily need to do what you did in undergrad. It's very, very open. Yeah. Any questions? Don't be shy. You don't want to know anything about, I don't know, about anything. How did I choose UC Merced? I think UC Merced chose me. I sort of didn't want to go to, I didn't even, I was this is the new UC, let me just apply, But I wanted to go to Berkeley and, but they offered me a lot of money. So they paid for my tuition and I had change, a lot of change. So, and it's a small school and, the environment is very peaceful. You see cows everywhere. And but they foster your potential. They really create a platform for you to thrive. I've been able to do research, to go to conferences throughout the country, and even being here because I met them at UC Merced. At UC Merced, I learned through the research programs. I learned how to be able to, I was dying, I was sweating, nervous, but I practiced from UC Merced. In a school like UC Berkeley, you will probably be a drop in the ocean, because there's so much potential, not only, but a lot of people. And to get research, you have to be, or know the professor, know the professor. But at UC Merced, it's offered, and there's a lot of funding. So yeah. And equipment, most of the things there are new. So yeah. I definitely encourage you to go to UC Merced. You won't regret it. Yeah. Or UCSF for grad school. There's no undergrad, but yeah. Yes?
What was the application of the stem cell research towards fighting cancer?
The application for this?
Yeah.
So, like I was saying, cancer cells are proliferative cells, right? But cells that differentiate and become organs or tissue, other tissue, they come from stem cells, whether embryonic stem cells or intestinal stem cells or epithelial stem cells. So all of these are coming from a source. If you understand the source and you understand how the differentiation process happens, you can manipulate it, right? If you master something, you can tweak it. So understanding stem cells and how they differentiate, how competition happens, how they're being replaced, because the cells that you were born with, you don't have them right now. They're completely replaced. So understanding how they're replaced, how many they make, all of that helps with cancer, because cancer cells are just cells that quickly divide. So why are they quickly dividing? What's in their environment? Are they coming from a specific cell? Is there a trigger in the source stem cell? So understanding stem cell is definitely important with understanding cancer and potentially finding a cure. Yeah, good question. Questions? You can ask, it doesn't have to be like research related or technical. You can ask about life, how hard it is to be a STEM major. It is, especially as a woman. It's extremely hard.
You said you grew up being interested in math and chemistry.
How was I interested in it? I think interest sometimes is sparked with being ability, being good at it. And math was good. I was good at math. And chemistry, I was good at chemistry. I tutored chemistry for a couple of years. I tutored math. So, I just decided to study that because I only had a couple of years, 2, 3 at most, as a transfer student. So I was like, I'm good at this, and with chemistry and math, I can branch out anywhere. So, yeah.
Are you using your chemistry knowledge?
I am not currently using my chemistry knowledge, but I would say that my training helps me think about the research in a different way than someone who will come from a biology major, for example. I have a different outlook because I wasn't exposed to it. It's a fresh set of eyes, and I can come, arrive at a conclusion or the problem from a different angle than someone who has a different training. Yeah.
What's the training of the other people in your lab?
So one was a biochemist when she was an undergrad. The other one did psychology and biology, the graduate student Sumitra, and then the research assistant, I think she did biology. Yeah. So I have chemistry and math, the odd one. Yeah. That's good. It's good to be odd. Yeah. Yeah. Okay.
So really quick before we finish, I should have gotten up and introduced Jocelyne because I met her at UC Merced in spring. We took a group of students on a trip, but a couple of them were here. I was busy talking to her before the talk started, and so Rance got to introduce her, and I should have, but we were so impressed with her, and she was so much fun, and so gracious, and so excited about our students, and so welcoming to our students, and she shared her story with us. We didn't have anything to do with her educational journey, but we were so excited and so proud and so happy, selfishly, for us when she got into SF, because that meant she was close to us. So we're super excited that she was here tonight. So I just wanted to give one more shout out and thank you for coming. She's super amazing. So just thank you. We do have a couple more minutes. So if anybody wants to come up and ask questions one-on-one, she'll be here. And there's probably extra food left over, so help yourself.